Today, I will be giving a brief to help better understand the COVID-19 vaccine. I want to start this off by saying that the intention of this brief is not to pressure anyone to get the vaccines. This brief is going to be talking about those things that need to be understood before we can actually even start talking about the vaccine itself. Let's start diving in. First of all, hi, who am I? Why am I giving this brief? Before I joined the Navy, I was working in the Yale School of Medicine in the Department of Computational Biology and Bioinformatics, and our research was primarily focused in the field of genomics, which is the study of all of the genes in the human body, collectively called the genome. And then I was also working on specifically some human genome privacy projects. Before that, I worked at Pfizer in their drug discovery group, and particularly with the high throughput sequencing folks. So that involved dealing with a lot of data, publishing protocols, managing a lot of compounds and data and protocols was primarily what my role was while I was at Pfizer. Before that, I was getting my master's in molecular biosciences and bioengineering. I was in a proteomics lab. As I said, with genomics, it's the study of the collection of all of the genes in the human body, which is the genome. And proteomics is the study of the collection of all of the proteins in the human body. And then we were taking proteomics and we were applying it to a lung cancer problem. Proteomics, as it turns out, was a really valuable field for me to have studied because a lot of the immunology relies on the interaction of proteins. Specifically, how the coronavirus hijacks our human cells relies on a protein-protein interaction between the spike protein and the ACE2 protein. We'll go into all of this, so it's okay if it doesn't make sense yet. Before that, I was getting my Bachelor of Science in Mathematics, and I got a minor in biology and a minor in anthropology. And it was that minor in biology that really inspired me to go on this path through the biological sciences before I joined the Navy. As part of my bachelor's, I was an undergraduate teaching assistant for a cell biology course. And in that cell biology course, they learned the foundations of all of the organelles in the cells. They learned how genetic information is turned into proteins and then how proteins do things in the cells. In my master's, I was a graduate teaching assistant where I walked them through an experiment and then we had them write a paper on it piece by piece. We taught them how to communicate scientifically with their peers, which is also a very valuable skill. So I give you my credentials not to say that I'm an expert by any means in any of these fields, but rather that I have enough of a foundational knowledge and I have enough experience to be able to read the scientific literature, digest it, and disseminate it to a wide range of audiences. By the end of this brief, you all can expect to learn some of the impacts of COVID-19 in the United States and worldwide and why a vaccine is necessary. We're going to talk about adaptive immunity, what it is, how it works, and the roles that vaccines play in developing that adaptive immunity. We're going to talk about the typical vaccine development timeline, and we're going to compare it to the Pfizer timeline. Next, we're going to talk about how the coronavirus specifically hijacks our human cells and turns them into virus-making factories. Then we're going to talk about the types of vaccines that are available and then what is in messenger RNA vaccines specifically. We're going to go over the Pfizer vaccine safety and efficacy. Then we're going to go over some common myths surrounding the COVID-19 vaccine. Lastly, we're going to talk about how to sort through COVID-19 vaccine misinformation when you come across it on the internet or out in the world. So first of all, why do we care? Well, as of March 18th, 2021, there were over 29 million COVID-19 cases in the United States. There were over 130 million worldwide, and all of these cases resulted in over 500,000 deaths in the United States and over 2.23 million in the world. These cases have also resulted in literal trillions of dollars being spent by the United States alone on COVID relief programs. There were also a lot of jobs that were lost as a result of this pandemic. Just in one single month in 2020, there were over 20 million jobs lost in the United States. In Asia, that number was around 80 million. Worldwide, it was unknown, but just with the United States and Asia, in those two regions, we lost over 100 million jobs, which severely impacted our economies. I'm sure, as all of you are familiar with, we also had to experience lots of travel restrictions and mandatory quarantine periods. People have had to quarantine for 14 days whenever they do have to travel. We've also had to isolate a lot, and with quarantining and isolation, we've experienced a lot of loneliness, and loneliness without a pandemic is already one of the greatest killers in the United States and worldwide. But especially with this pandemic, we have seen substantial increases in both suicide ideation and deaths by suicide, particularly among young adults and teenagers. And with all of these jobs that were lost, and as economies have slowed or come to a complete stop in some countries, we have experienced a global recession that rivals that of 2008. So it's really important that future COVID-19 cases be prevented. And while we can wash our hands and we can socially distance and we can wear masks, medically, the only treatment that provides preemptive protection from viral infections is vaccines. But in order to talk about vaccines, we first have to talk about how adaptive immunity works from a very high level. So it starts off with a macrophage. And the macrophage, it's a white blood cell. And its job as a white blood cell is to patrol the human body looking for something that causes disease. For example, a virus. The more generic term that we use for something that causes disease is a pathogen. And what happens is the macrophage, it patrols the body looking for a pathogen. And when it comes into contact with it, it doesn't know what to do right away. So it picks up the pathogen and it breaks it down into its protein components. And then it presents those protein components to a second type of white blood cell called a T lymphocyte or a T cell. Now this T cell, it's going to do one of two things. The first thing that it can do is it can turn into what we call a killer cell or a killer T cell. Killer cells or killer T cells, what they do is they patrol the body looking for human cells, and they look for human cells that have been infected with the pathogen. And the way that they can tell that the cell has been infected with the pathogen is because the proteins that the T lymphocyte were presented with are on the pathogen that are coming out of the cell. So it recognizes those proteins coming out of the cell on the pathogen and it destroys that cell. Then the remnants of that destruction are digested by the macrophage, that first type of white blood cell. So that's the first path that this T lymphocyte can take. The second path that it can take is going on to activate something called a B lymphocyte or a B cell. B lymphocytes, they're responsible for making these little Y-shaped guys called antibodies. Antibodies are a very specific defense mechanism against the protein that was presented to the T lymphocyte. The tips of these antibodies, the tips of the Y shape, they will change their shape depending on what the shape of the protein is. So I'm going to do a demonstration here. If the protein has a shape that looks like this, the antibody, the tip of it, is going to have a shape that's perfectly complementary to it so that it can bind very tightly to the protein on the pathogen. And that very tight binding is crucial because of two things. The first is, let's suppose that the protein on the pathogen is responsible for binding to your cells to hijack your cells and then turn them into virus-making factories. If the antibody is binding to that binding site on the protein on the pathogen, it's blocking that binding site, and now that binding site can't be used to bind to your cells to hijack them. So that's the first potential benefit if the antibodies are targeted to that specific binding site. The other thing is when enough of these antibodies are sticking to the protein on the pathogen, the tail ends of these antibodies are made of sugars, and that signals to the macrophage, that first type of white blood cell, to come and digest the pathogen that's been covered with antibodies. So that's a really powerful way that our bodies can fight off pathogens, or in this case, a viral infection. But the thing is, to get that perfectly complementary shape to the protein that was presented to the T lymphocyte, it can take a long time. It's something on the order of 10 to the 8 different combinations that the B lymphocyte has to go through before it gets those antibodies that are perfectly complementary to it. Once it figures it out, it's going to create these plasma cells that produce those specific antibodies. And on top of that, our bodies want to remember how they created that perfect shape. Our body makes something called memory cells, which retain the information to make that specific shaped antibody that's complementary to the binding site of the protein that was on the pathogen, which was then presented to the T lymphocyte. The reason for that is so that if you get infected with the same virus that has the same proteins on it, your body can respond to that threat right away. And that's really powerful.